This is Homeschool 101, a collaboration between ABS Television and the Ministry of Education. Stay attentive and attempt all activities. And now, today's lesson.
Good day all and welcome back to CSEC Biology. We're going to be continuing with the topic genetics. Remember, genetics tells us who we are and how we function, how we look and how we behave in different situations. All right? Now what we're going to be doing first is we're going to be recapping some of the terms and some of the concepts that we would have completed last week in our session. We looked at several key terms, two of which were the phenotype and the genotype. And the phenotype is that particular trait that we see on the outside. The genotype is the blueprint that tells the phenotype how it should look. Two other terms that we looked at were homozygous and heterozygous. Remember, alleles appear in pairs. That is how the genotype is written. And so there are homozygous, meaning that the alleles are the same, as in we have two uppercase letters or two lowercase letters, that's homozygous, and heterozygous when we have an uppercase letter and a lowercase letter. And the last two were dominant and recessive. Now remember, dominant traits are those that are seen in the actual phenotype of the individual. And the recessive are those characteristics that are only seen when two recessive alleles are paired together. Another thing that we did was we mimicked the genetic inheritance process, and we used the two tall individuals where both of them were heterozygous for the tall gene, and we mimicked that by using red and white beads that we were able to place in two containers and pick, and we were able to actually record them in a data. And we had a bit of a teaching moment in terms of how to present information, because on CXC we have to remember that our table borders must be all present, all four sides must be present. Our table must always have a title, and this title must be placed at the top. We must remember always that our column headings and rows must be filled with the correct content and headings as well. We also looked at a gentleman by the name of Punnett, and he developed this square that enables us to predict the inheritance of genetic information from parents to the offsprings. And one example that we used to illustrate the use of the Punnett square was the tongue rolling. Remember, one of the activities that you could have engaged in was to go home and ask your parents if they're able to roll their tongue and watch them do it, and then you are able to use their genotype and figure out what the possible offsprings could be. Now, we're going to be using a bit of the multiple choice in order to help us remember some of the concepts that we learned last week. Now, if you know, the exam this year is going to take the form of a multiple choice paper along with the SBA component. And multiple choice questions, you're either right or wrong. So we want to make sure that we have the skills that will enable us to identify the true correct answers. All right, some of the questions may be a little bit on the tricky side, so we're going to have to be able to learn some strategies that can be employed so that we can get the best out of each question. Now, the first question is, an allele is a damaged gene, one form of a gene formed by genetic engineering, a gene that does not have a recessive form. So I'm going to give you like 10 seconds so that you can try and see if you can get the answer. So an allele is one form of a gene. Remember, we looked at genes are basically characteristics, and some of these characteristics can have a dominant or a recessive part to it, and those parts are called the alleles. Next question. A recessive allele is a form of a gene that is always the cause of harmful features, is never shown in the phenotype, is not present in the heterozygous state, or only shows in the homozygous state. And I give you like 5 seconds. 10 seconds was a little bit long. So 5 seconds, we're going to try and identify the correct answer. All right, so the answer is D. The recessive alleles are only shown in the homozygous state because remember, they are recessive and they would only be phenotypically represented when both the alleles come from the recessive. All right, number 3, which is a true statement about genes. Genes are built from 2 or more alleles, are linked to form pairs of alleles, are only present in dividing cells, or control the inheritance of features. All right, so which is a true statement about genes? D, they control the inheritance of features. And I want to stop on this question a little bit because it could say which is the correct statement. So we have to bear in mind some of the key words that are going to be used in the questions themselves. If you're not truly understanding what the question is saying, maybe you need to read it more than once so you can grasp what it's actually saying and you can identify the correct answer. All right, so the unit of inheritance is a chromosome, dominant allele, gene, recessive allele. And the answer is a gene. Gene is that thing that gives us tells us who we are, gives us the information of how we are going to look and how we are actually going to be behaving as well. Now, the last question we're going to be looking at today, different forms of a gene are known as A, alleles, B, gametes, C, genotypes, D, heterozygous. And the answer is different forms of a gene are known as alleles. All right? I do hope you were able to identify at least 3 of the answers and get them correctly. Now today we're going to be starting a new arm of genetics, and here is a picture of a cow, of 3 cows. They are, or calves, they are different colors. The first one, which is here, is black and white. This one in the middle is brown and white, and the last one is black and white as well. Now, can you guess what color their parents would be? So we're looking at the inheritance of the fur color in cows. All right, now more than likely their parents would be black or white. Well, one will be black or one will be white, or they could look exactly like the calves here, where they would have a combination of both colors. This is an example of codominant characteristics. Now, these are traits that we see where there is no dominant recessive. Last class, we looked at dominant and recessive. However, there are inheritances where none of the characteristics or none of the alleles are dominant over any one. So we have both of them being represented in the actual phenotype of the offsprings, where we can see here. So more than likely, this first cow had a white and black, a white parent and a black parent. This means that both the alleles are represented in the phenotype of the offspring. Now, we can look at the Punnett square and we can see how we can try and figure out what the possible offsprings could be, or what the possible parents could possibly be when we look at the inheritance. So last week we learned about the Punnett square, and we're going to incorporate that into our lesson today as well. So remember, there is no class or no concept that is taught in isolation. Everything is going to connect with each other so that at the end of our 5th year we would have a combination of information that would just lead into each other. So we're going to draw our Punnett square. Remember, the Punnett square is just a square with 4 small squares in the middle. And the homozygous, so if both parents were homozygous, there would be WW, that would represent the white, and there would be black, which would represent the black. Now, when we're looking at codominance, we always write the genotype with both uppercase letters. Our last session, we looked at uppercase and lowercase. We were dealing with dominant and recessive. However, today, we're looking at codominance. And in codominance, there is no one characteristic that is going to be dominating the phenotype. And so both of them are going to be written with capital letters. So we will do this. And we know that that would be BW, BW, W, and BW. Now this could be the cow that we saw earlier, that first cow, who seemingly had a white coat with black splotches on it. All right, it could be either one of these offsprings. Now we can also look at the heterozygous. So their parents could be heterozygous, meaning that they contain both of the alleles. They contain the black and they also contain the white. And we can do the same with that in terms of using the Punnett square to illustrate the offspring's genotypes. So we'll draw again. And remember, when you're drawing, you're going to try and always use a pencil and a ruler to assist. When you hear the word draw, we automatically think about a pencil, never a pen. So we're going to look at BW, and we have another BW on this side. And then this organism would be totally black. So this calf would be homozygous for that black trait, that black coat. And these two offsprings would more than likely be the two calves that we saw in the picture, in the previous picture, where they have a combination of black and they have a combination of white. And then WW would mean that there may be a possibility that one of the offsprings could be totally white. All right? And we looked at the percentages and so on last class, so we're not going to go too deep into that today. Now, this is another example of the codominance. We see it in cattle as well. One is white and one is red, and you see the splashes of red that are on the cows. All right, so the genotype in these allele combinations are always written with the 2 uppercase letters, as I mentioned before. Both alleles are expressed in the phenotype of the organism, so you're going to be seeing it on the outside. One very good example of codominance in humans is the blood group AB, and we're going to look at that in depth. Now, if you look at the blood group AB, you know that there are 3 possible alleles. There's A, B, and O, and they can come in various combinations. All right. So A and A would make 2 double A's, and this is the one that we want to really focus on, the AB. Let me just put that here. Now remember, when both the alleles come together and they are seen in the phenotype, it means that they possess a type of codominance. And that's what we see when the allele A comes together with the allele B. There's no one that is actually dominant over the other. And so if you have the genotype AB and you go to the medical lab to get some tests done and they pull your blood and you want to know what your blood type is, your blood type will more than likely be AB. So your genotype and your phenotype is basically the same, and it's representing both of the alleles in the actual phenotype. All right, so this is another example of how we can actually get it using the Punnett square idea. So if one of your parents has the genotype AO, which would mean that their phenotype would be A. That would be their blood type. It would be A, depending on whether you have a certain antigen. And they will get the positive or the negative, but we're not going to go into that today. And if another one of your parents has the genotype BO, then we know that the blood type that will come with that would be B. All right, so when we bond those two together, here is where we're seeing the codominance, where both of the alleles are represented. But you may be wondering, what about the others? What about this AO and what about this OO? Now, AO, there is a type of dominant recessive type of thing going on with the blood groups as well. Because when AO is paired together, O acts in a very recessive manner. So you will never see that in the phenotype. You would only see A. So if your genotype is actually AO, when you go and you take that blood test and they take your blood and you're trying to find out blood type you are, it is always going to be A. It will never be AO because O has this type of recessive behavior when it is paired with either A or B. However, when it is paired with O, the blood type that we get is O. And, the antigen would tell us whether we are positive or negative. All right? Let's move on. Now, that was codominance. So remember, in codominance, we have both alleles being represented in the actual phenotype. In incomplete dominance, it's slightly different. Incomplete dominance means that you're not actually seeing the two individual colors or the two individual features. And it's very, very evident in this example that I have on the board here, where we have a dark and a light, which is white, giving rise to a type of gray color. All right, and that's a combination. And then we will see the F2 generation. But we're not going to get into F2 generation. We're just going to stay right here on the F1 generation. And we look and we see that all of the offsprings are going to be a type of gray color. And that is incomplete dominance, where the alleles are like mixed together. Okay? We can have a more visual one in terms of the flowers. Now, in codominance, this is codominance and incomplete dominance in flowers. So if you notice, these are the two parents at the top right here. There's a red flower and there's a white flower. And in codominance, you see that the offsprings would have a type of white border around the petals, and then the inner petals would have this red color. That is codominance because both the colors are going to be represented in the phenotype. However, in incomplete dominance, we notice that the offsprings would be pink. So the red and the white would mix together and they would produce this beautiful pink color. And it is here in the Punnett square. You can see an illustration of that. Now, in Antigua, we have bougainvillea. I don't know if you know what bougainvillea is, but it's all over the island. It's all over the island. And this is a very prime example of incomplete dominance. And you can see it in the photograph. There are some red bougainvillea flowers and there are some white bougainvillea flowers. And they got hooked together and they produce a lot of this pink color, which is a very good example of the incomplete dominance that we can see inside of actual flowers. And there we have a Punnett square that enables us to see. So when the heterozygous in the codominance mate together, we get a combination of red, pink, pink, and white. And it's represented here in the photograph. Let us just do a little bit of recap. So there's codominance and there's incomplete dominance. Incomplete dominance means that there is no one trait that is seen in the phenotype, but it's a combination of the traits. So white and white would make pink. That's incomplete dominance. Codominance means that both of the alleles or both of the characteristic traits would be seen in the phenotype. So you would see a red flower with white guidelines. And you know what the guidelines are in the flower. Those are the little lines that guide the bead towards that area that it will be able to begin making its honey. All right, so the flower may be white, but there's going to be some red guidelines. Or the flower may be red at the tips of the petals, but have white in the inner petals. All right, and that is an example of codominance. Now we're going to move on to variation. And we're a tropical island, so I have some varieties of mangoes that I was able to collect actually yesterday. I wanted to get them as late as possible so they can be as fresh as possible. I want to ask you one question. Which one would you like to eat? If you were in the studio with me right now and I ask you to choose one mango that you wanted to consume from this table, which one would you pick? Of course, everybody's going to pick the big one, which is this one. It's big. It's going to have on lots and lots of flesh, and it's going to be satisfying for not only one person, unless you just want it for yourself. But why? Why is it that all of these are mangoes? However, they're different. Because there is what we call variation. There are differences among species that make them the same, but make them also different, all right? Their characteristics. We're going to be using variation in humans, and we're going to be looking at some examples right now. Before we get into the actual examples, we want to know, what is this variation? What is it? Variation is the differences between individuals within a species. And this is a prime example. These are mangoes. And there's so many different ones. This one is shaped very oval. This one I've never seen in my life before. It's very, I don't know what shape you would call that. This one is different. This one is large. This one is, I think, a kidney, a little rounder. And this one shaped almost like a little square. All right. Now, variation can be seen in individuals, not only in the fruits that I have on the table today. We can see variation in terms of height and in terms of the skin tone of our people. This can be caused by inheritance, which is gametes who are going to bring genetic information when you are being formed. And also environmental factors can also play an influence in how we actually look. Variation has two basic types. They're continuous and discontinuous variation. And we're going to go into all of that today. Now, continuous variation, continuous. Play off of the word, it's continuous. Continuous. Human height is an example of a continuous variation. It ranges from that of the shortest person in the world to the tallest person. Any height is possible between these values. All right, so it is a continuous type of variation. So we don't have a system where you have to be 6 feet tall, 5 feet, 4 feet, 3 feet. Everybody doesn't fall into those 4 categories. And that is why this type of example is continuous, because you can be 5 foot 1.5, 5 foot 3/4. And there's so many values that can be between 5 and 6. All right, so we say that it's continuous because there's no definite group that everybody would fall into. And you see the photograph. We look at the skin tone. There is no light and dark. There's shades. We are shades. And we can see it going from the darkest to the lightest. There is also discontinuous variation. Now, this is one that I really like to do with my class. We would get some type of ink. And I would suggest, since you're home, you can use beet. You just peel the beet, and then it's going to give off its pigment. And what you do, you just go around the house, and you just press your finger to the beet, and then you press it on the paper. And you'll be able to see your fingerprint very clearly. Use only one finger, though, okay? So discontinuous variation, a characteristic of any species with only a limited number of possibilities or values, and this is what it shows, the discontinuous variation. Now, as I said, I use the fingerprinting thing. We can actually try this at home using the red pigment found in beads, and you can use your index finger. Try it with the entire family to see which pattern do you have. Do you have the loop? Do you have the whirl? Or do you actually have the arc? And there's actually another one called the compound. And every year I do this with my students. I have never seen a compound individual. So that's one of the reasons why I didn't include it here. All right, but you can go online and you can see if you can find that compound fingerprint pattern, and you will be able to see, oh, I am a compound, or I am a loop, or I am a whirl. All right? Now, these are some examples of the 2 types of variation, all right? Here are some examples of variation that you can investigate in your family and your immediate environment. So remember, we are living in a very serious time, and so we can't venture out too far from home if it's not necessary. So go out in your backyard. I'm sure you have trees. Look at the leaves. Look at the size of the leaves. Look at the shape of the leaves. Look at the height of the individuals that you see on the road. You don't have to have the exact. Look at the skin tone. We looked at that one. I looked at the leaves. I spoke about the leaf size. Tongue rolling, which was one that we did last time we had this session. Look at whether the leaves are plain green. I'm sure you've seen some leaves that have some beautiful colors like yellow and red and so on. Look at, it's either they are variegated or they're green. There's no in between. You can't sometimes roll your tongue and then sometimes not. You either can or you can't. Horns in cattle. So while you're driving, you can look at the cattle and see how many of them may have horns and how many may not. Earlobes, and that's a very, very common one, because you can do that with the people in your homes. There are attached earlobes and there are free earlobes. The free earlobes are the ones that dangle, and the attached are the ones that actually kind of form themselves onto the face of the individual. Hand clasping. Now, I like to do this one in my class. I would go around and I would ask the students to just clasp their hands, and they will clasp them. This is how I clasp my hands. So we would look at whether or not you clasp your hand with the right thumb on top or the left thumb on top. Now, for me, I clasp my hand with the right thumb on top. If I try it the next way, it feels kind of funny. It can happen, but that's just my preference. And we can also venture into the toe length, and definitely you can do this in your family, because they won't be afraid to take off their shoe and show you their toes. So we're going to look at whether the second toe is longer than the first toe, or the big toe. And I have some pictures so that you can see. All right, so we know the height, basic. Skin tone, we know it moves from dark to light. And the leaf size. Look at these different sizes of leaves. There is no rule that says all leaves must be 5 by 6 or must fit into this mold. There are many different sizes. If you even look at a banana leaf, that is huge. All right? Let's move on to the discontinuous variegated leaf. This is one of the flowers the plants that I was mentioning earlier. It does have green, however, it has some beautiful red, royal red, if that's even a color, combination here. And then there's the regular one. So it's green, or it's going to have a combination. It could be white and green, it could be red and green. Or it can be other colors, it just depends on what species you're looking at. And the discontinuous type of variation is the earlobe. And I'm not sure if the camera can zoom in a little close, but you can see these are the free. The free earlobes are the dangling ones. And the fixed or the attached earlobes are the ones that come straight down and attach themselves to the face, actually. The second toe length. Now in this first individual, the second toe is very long above the big toe or the first toe. And in this one, the second toe is shorter than the first toe or the big toe. And there's also, if you want to go a little bit deeper, we can also go into the shape of the toes. This first one is that slope straight across. I've never seen anybody with their toes like that, however. But then again, you don't see everybody's toes that you encounter. This is one that we looked at because remember, we looked at the second toe being long, and this one I've never seen before. It's like the first 3 toes are on the same height, and then the last 2, they just are very short. All right, so that's discontinuous variation. So we're going to summarize today. Now you have a wealth of information about genetics. You can go into your homes and you can investigate your parents, and they have to put up with you because you're their child. So you use these terms, you use these concepts, you use these characteristics to help you to learn about genetics. Because genetics is so relatable, we don't want to have to sit down in the book and study, when once we can apply the concepts, it becomes a part of our daily life, and it doesn't need to be constantly put back into our minds, okay? You're equipped with this new knowledge, so you can go forward and you can master your exams when they come around in July. Thank you for joining me in this session, the second session of genetics, where we discovered lots of things about why we are the way we are and how we are the way we are. Go forward and master the exams when they come around, and goodbye and all the best.
You've been watching Homeschool 101, a partnership between ABS Television and the Ministry of Education. Thank you for viewing this lesson. Join us again next time.